SNU, all participants, my colleagues and friends. On behalf of the member of Central Calcutta Society for Advancement of Human Development and Research, I welcome you all to the National Symposium on Combating Human Diseases, Quest for Interacting Natural Products, organized in collaboration with Indian National Science Academy and the Department of Microbiology and Biotechnology of this university. By now, I believe everybody are aware that the genesis of this society started at 1993 and its function and goals. We organized the annual day-long symposium on different themes based on contemporary scientific issues related to human development and health, where physicians and scientists share thoughts and get involved working towards betterment of human health. This time, we selected this topic especially to commemorate the birth centenary of Professor Ashima Chatterjee, whose pioneering work on secondary metabolites of medicinal plants paved the way to present-day phytochemistry and phytomedicine research in this part of India. Through the use of medicinal plant products dates back to time immemorable, the search for actual component derived from natural sources and its mode of action in human diseases gained momentum rather recently. And with the help of modern technology, production of phytodrugs and its delivery to the active site attained tremendous interdisciplinary research interest. We have with us experts from the existing area, an exciting area of research, and I'm sure the deliberation would enrich us with new knowledge. I myself am very happy to see many of our former Foundation Day oration and Professor K.L. Mukherjee Memorial Lecture awardees participating in this meeting. I wish you all to have a pleasant day with lively discussion. Thank you. I now invite the Vice Chancellor of this university, Professor Dhruvanjyoti Chattopadhyay, to deliver the inaugural address.
A very good morning to all of you. Today, we are here to commemorate the birth anniversary of Professor K.L. Mukherjee, who is one of my teachers in my biochemistry classes. And he actually taught us the relationship between the clinicians and the biochemists. I still remember the way he used to teach us. It's something very difficult for us actually to first to digest and then to understand that, oh, what it is. And that we are going to, during our general viva, we call it grand viva, when sir used to take the grand viva, from that day we understand that why he used to teach us in that way. With each practical applications of biochemistry, in the health science research. He coordinated in such a nice fashion. That is something which is very, very important. Now today, what we are going to do, we're going to celebrate the 32nd Foundation Day celebration of this Central Calcutta Society for Advancement of Human Development and Research. And we are really fortunate, and we are very, very thankful to Professor Ashima Lahiri Mujumdar and her team for identifying Sister Nivedita University as one of the venue for organizing this particular symposium for the last 3 years. Thank you, madam, for actually identifying our university, because that is going to help quite a bit to our faculty members, the students, and also this poster session which I could see, whatever we could see in the first year, in the second year it again, we have seen the how number of posters increased, and then this year I heard, after third year also it's quite a bit, and this year we heard that there are a large number of posters and we cannot accommodate every one of them. So which shows that yes, among the young budding students who want to do research in the area of allied health, the importance of this particular symposium is becoming more and more. I also want to thank our collaborating organizations like the Indian National Science Academy, where Professor Arun Lahiri Mujumdar is actually also a senior professor in our university, is helping us to organize this particular program. I want to thank our mentor and the founder, Vice Chancellor, Professor Ashok Thakur, who actually started this particular university as well as the department also. And sir's vision is always something which helps us to go forward. The other personalities who are present over here, my friends, the esteemed speakers who are present over here, and the faculty members, students, researchers, and the other speakers. One thing is very important, that the topic of the symposium in each and every year, it's being identified in such a way so that it is related with something where we can actually unravel a lot of new important informations. And as Ashimadi already mentioned, that we are celebrating the Professor Ashima Chatterjee's contribution in this natural products in this research area. So from that point of view, this seminar is going to be very, very important. Now, what you can see, the symposium is on combating human diseases, quest for interacting natural products. And rightly mentioned by her is that it's from the, I think it's already for a very, very long period of time, we know about this importance of the natural products, but our concept regarding these natural products are changing quite a bit. Because at the very beginning, the natural products are looked like that over here, we looked at it from a completely different point of view, but now people started to look after the different types of, say for example, from the Egyptian medicine, from the Chinese medicine, from the Indian medicine heritage like the Shushrut and Charak and Susruta, we could find it out, the importance of this particular medicinal plants in combating different types of researchers. And when we are talking about this, combating this diseases, it's not only the communicable diseases, but the non-communicable diseases also. Say, for example, when we looked at it from the point of view of how we actually right now the point is here in the type 2 diabetes research, there are quite a bit of natural product related compounds are being identified, which played very important role in these treatments of this diabetes, type 2 diabetes. And there are a number of papers which are coming out right now. So what we can see, that importance of these natural products as medicine, as products which are going to combat, which are going to help us for in this health science research, is continuously growing. And it's not in just identifying a chemical, or it's just identifying some products in combination of other products, but every time we are getting now the new branches of chemistry, this particular drug is actually there with respect to the computational chemistry. Using that, looking at this particular chemicals from the structures by QSR analysis, we are getting new products also. And so it's continuously increasing, and I'm sure that today the different personalities who are going to deliver the lecture, from them our students are really going to be benefited about what's going on in the research area on these natural products. So with these few words, I really want to thank all of you who are present over here and who are going to join us afterwards for making this particular program a success. But to my dear students, I just want to mention that it is very important that in our university, the biotechnology, microbiology, pharmacy, the other health sciences like the biomedical, medical laboratory technologies, operation theatre technologies, all these areas are under one particular school, what we call the School of Allied Health Science and Translational Research. So why we put this name is actually to give some importance to this whole area, where we can see that the whole research what we are doing, whatever knowledge we are gathering, this should be translated and is going to be helpful for the mankind, and it will definitely going to help the society itself. So I really want to thank all of you and wish a great success of this particular program. Thank you. Namaskar.
Thank you, Professor Chattopadhyay, for your excellent inaugural speech to inaugurate that event. I request Rajat to come on the stage and give his vote of thanks.
Honorable Vice Chancellor, Professor Dhrubajyoti Chattopadhyay, honorable speakers, guest of honor for today, Professor Tulasiram, dignitaries, and all the students. Today, as told, that for third time we are organizing this one-day national symposium in our campus. And I hope that on the next years also we shall get the opportunity to host this seminar or symposium in our campus, and my students will be privileged enough to present their works in this auspicious occasion. So now at the end of the inaugural session, I am thanking all the participants. First of all, I should thank Professor Chattopadhyay for giving us the permission for organizing this seminar here. I thank Professor Tulasiram for accepting our invitation for coming here, and also all other distinguished lecturers, speakers, and chairpersons. It's a huge list, so I'm not taking the names of everyone. All the faculty members of Microbiology and Biotechnology Department. A very special thanks to ALM sir and ALM madam, because always they are a very big support for the society and our university as well. And Dr. Kajuri Lahiri, the secretary of our organization, Dr. Shom Shubro Thakur Chaudhuri, the treasurer of our organization, And I'm not thanking the students and the volunteers, but still I appreciate their hard work. And the participants, I hope you will enjoy a lot and you will listen to the lectures and you will gather a very good amount of knowledge from this event. Thank you very much. So one little announcement. Now there is a tea break. So I'm requesting everyone to just go outside. My, I guess website, sir. After the tea break, sir, the website will be opened. Yes, the stations, and before that, the website will be opened. So my volunteers are there. Please, volunteers, assist everybody to go to the area where the tea has been served. Please, everyone.
We are gathered here to start our award session for the event. May I request Professor Dhrubajyoti Chattopadhyay and Professor Mitali Chatterjee to come on the stage and chair the session. Madam. I take my immense pleasure in inviting Professor Chattopadhyay to introduce our distinguished speaker for the Foundation Day oration, Dr. H.V. Tulasiram, Principal Scientist, National Chemical Laboratories, CSIR Pune. Sir, please felicitate him with Uttariya.
We are really very happy to have the Foundation Day oration speaker, Dr. Tulasiram. Dr. Tulasiram, who completed his MSc in chemistry, organic chemistry, and then PhD in bioorganic chemistry from Indian Institute of Science, Bangalore. Then he actually gone abroad for postdoctoral training in University of Iowa, and followed by University of Utah. In 2006, he came back to India and decided and actually joined the position in Natural Product Research at Division of Organic Chemistry in the CSIR National Chemical Laboratory. And till date, he is working over there and established a very, very vibrant research group to work on chemistry and biology of natural products. Now, as natural product plays a critical role in the human well-being and the levels are very low in natural sources, Professor Tulasiram established a state-of-art laboratory and expertise to carry out the metabolomics and systemic dissection of biosynthetic pathway for the value-added molecules from traditionally and medicinally important plants, with an ultimate goal of producing these molecules and their derivatives in the metabolically tractable heterologous system. And it is very noteworthy to find that during this short period of time, 18 PhD students are graduated and another 4 students are pursuing PhD under his supervision. During this period of time, he published more than 90 research papers in various internationally peer-reviewed journals and has got 8 patents. His group has identified and characterized over 180 novel genes involved in isoprenoid biosynthesis. May I now request Professor Tulasiram to come over and deliver the Foundation Day oration lecture on chemistry and biology of neem limonoids. Professor Tulasiram, please, let's give a big hand to him for this.
Indeed, it is a great honor. Professor ALM called me. Tulsi, you should come for a seminar. I surprised of what to give in the human diseases. Because after coming here, I noticed that. So we work on that natural products. And as the title says, quest for interacting natural products. It is necessary to, it is audible now? Yes. Okay. So interact natural product finish along with biology is very much necessary now. So before getting into my talk, I should thank Madam Helen, Sir Helen, and Rajat, and Sir for good, nice introduction. Allow me to- even though I don't know whether I deserve that. So I am really happy to be here and all the organizers, Rajat, Professor Rajat, for calling me and asking me to give the talk. So the natural products or medicinal plants is used in Ayurvedic medicine and potent insecticidal activity, especially anti-fungal activity. Similar way, there are several plants. Take few examples. Difficult to find that. Yeah, as a farmer, yeah, last week I was in my village. The sheep got completely into I asked the village person here. He got some rocks and he report me. I apply. It is really healed that damaged. No, it is working now. It is working now. Damaged eyes. But when I try to get what is the plant he used, he promised, it seems, he should not tell. He should pass it on to another. This is how things are going away. Even though we have very good resources, difficult to get same thing. Same thing, another example if I want to give, Lucas aspera, that tumbe we call it, Lucas aspera, that plant, the flowers, whenever somebody has a high fever, they crush with a along with jaggery and give that peak moment. Next time they won't get that fever again. But when we started isolating molecules from only flower, it did not give any activities. So what we have to do, the formulations we have to take and analyze the individual component and study their activities. Then only we can get something out of it. So with this, today what I am going to present is our studies, last 15 plus years of studies on neem limonites. So because neem contains major compounds, limonites as a major compounds. So what happened? F5. All these guys, several research associates and project assistants, and of course funding from CSIR, DBT, ICR, ICMR, and direct CSIR and CR is highly acknowledged during this talk. Okay. So the major focus of our lab is study of natural products with biology and chemistry aspects. In chemistry, we isolate the molecules, identify, characterize them, and then look for the collaborators for biological activity. In biology, we isolate, do the transcriptome and genome studies, and isolate the genes and characterize them, do biochemistry, protein engineering like mutations or site-directed mutations or the random mutations, check the activities, and also study using the labeled molecules, substrates, for their enzyme activity or mechanism of catalysis. That's how overall work. What it is a multidisciplinary area, and once if we identify, generate this much knowledge, our aim is to produce these molecules, as Sir said, metabolically tractable heterologous system, whether yeast, bacteria, algae, or so on. So we are working on several plant systems. As you can see that neem, sandalwood, Cathananthus roseus, Andrographis paniculata, Vedalia pulidosa, Osimum, and so on so forth in biosynthetic pathways. And also once we isolate the molecules, we modify them using organic chemistry, synthetic chemistry methods, or using the biocatalyst, and see that their activity, biological activity, and what type of transformation it occurs, all those things. And as I mentioned, it is a highly multidisciplinary area. It uses concepts, knowledge, and tools of organic chemistry, biochemistry, molecular biology, bioinformatics, microbiology, and so on and so forth. It is a extensive crosstalk among the groups is required if you work on this type of research. So neem is everybody knows that it is used even anything in villages, even they put it in a boiled water and take the bath because if any skin disease or anything is there. So it is a wonder tree. It has several molecules which are biologically active, and over 250 secondary metabolites have been isolated, and among them over 150 molecules are liminoids, which are structurally complex, highly oxygenated, and several chiral centers, and it is very difficult to synthesize using organic synthetic methods. So these liminoids can be classified as a basic liminoids, as I don't know. Okay, basic liminoids, azadirone skeleton, guanidine skeleton, villacinine skeleton, and so on so forth, azadiractin skeleton, and all these molecules belongs to isoprene category or class. And isoprenes are major class of natural products. So nimbin is the first one isolated by Professor Siddiqui in 1942, and hence the liminoids later named them as a liminoids. So coming to isoprenoids, all these isoprenoids, since I am going to talk, my talk is divided into three portions. One is chemistry, where we isolate the molecules, modify them, and using them establish the analytical method, and using this method, the chromatographic method will be used for metabolic studies, and tissue specific levels of these metabolites will be analyzed, and then based on that knowledge we'll do transcriptome analysis, and the metabolite level as well as transcriptome expression levels were matched and try to identify the genes. That is the third portion, biosynthetic pathway. In the middle portion, after isolating over 30 liminoids, we approached several groups across India and studied anti-cancer, anti-diabetic, all those work. That is the middle portion. I will just scan through them. So over 80,000 isoprenoids are present. So that involves in that delicols, prenylated protein, carotenoids, monoterpenes, sesquiterpene, ubiquinone, all are involved in one or the other biological activities, and all these molecules are synthesized from two C5 molecules, that is isopentenyl diphosphate and dimethyl allyl diphosphate, that allylic diphosphate and isopentenyl diphosphate. These two molecules are involved for the generation of these many molecules. So these two molecules thought to be biosynthesized through MVA, melanate pathway. All of you studied, I think we take statins, which blocks MVA pathway for the production of cholesterol. So this MVA pathway thought to be universal pathway before 1990s. So in 8 steps, these 3 molecules of acetyl-CoA combine to form the isopentenyl diphosphate and then isomerize to dimethylallyl diphosphate in last step. So this pathway, also called HMG-CoA reductase pathway or melanate dependent pathway or isoprenoid pathway, so present in archaea, plants and all animals and fungi, especially eukaryotic cell. So till 1990s, early 1990s, Professor Romer, he analyzed all the feeding experiments. There is this discrepancy when they checked with bacterial biosynthetic pathway or plant biosynthetic pathway. Then he carried out several feeding experiments and found that there is another pathway exists, which is called methyl erythritol phosphate pathway or Romer pathway or non-melanate pathway, which leads to formation of dimethylallyl diphosphate and isopentenyl diphosphate in bacteria, plant plastids and malarial parasite and most of the bacteria. Hence it is a antibacterial targeting pathway also it is called. So in this pathway, the glyceraldehyde 3-phosphate and pyruvate. I have written structures because if you see the structures, because most of the students are biology here, if you see the one step to another step, what exactly happening you can recognize. That's why I never use the names. Names you can use easily. So, but to understand the exactly what is happening, same thing it is happening nowadays. We do PCR reaction or we do the RNA isolation. We will not be knowing what exactly this step does. Protocol, take that protocol, add this 3 microliter, 5 microliter and get the RNA. No. We should understand what exactly happening there. So likewise, I insist most of the, we can do, we can write, we can understand. If you write a structures, it is easy to understand each step what exactly is going on. So in 7 steps, both the molecules IPP and DMAPP formed in a methyl erythritol phosphate pathway. So there is a phosphoromycin in 1970s they used to use as an antibacterial agent. Till this pathway established, they did not know what was the target. It blocks the DXR enzyme where MEP formation is blocked. Same way in previous one, I'll just go back. Okay, sorry. Okay. Here the HMG-CoA reductase, HMGR, statins blocks this step to stop the formation of MVA. That's what statins we take when we have a high cholesterol. So in once dimethylallyl diphosphate and isopentenyl formation diphosphate formed, they undergo chain elongation, that is regular connection or regular chain elongation reaction or 1'4 condensation. This is a most common in most of the living systems. So one dimethylallyl diphosphate condensed with 1 molecule of IPP forms the geranyl diphosphate, and geranyl diphosphate condenses with 1 more molecule of IPP forms the farnesyl diphosphate, and which can again undergo 1 more IPP condensation to form the geranyl diphosphate, or farnesyl diphosphate can undergo the 1'1 dimerization to form the squalene. From squalene, it leads to formation of triterpenoids or steroid pathway. Whereas geranyl diphosphate, we use that carvone, menthone, menthol, monoterpenes are formed from geranyl diphosphate. Farnesyl diphosphate goes to artemisinin, potent antimalarial molecule, or the nice sandalwood fragrant xanthalols, or caryophyllene, humulene. So that goes to farnesyl type. And then the diterpenoid, where C20, monoterpene contains C10 molecule, C10 carbons or 10 carbons. The farnesyl diphosphate contains 15 carbons, and gernyl diphosphate contains 20 carbons. That is a diterpenes. So which I can go to Taxol, that potent anti-cancer agent. So this whole pathway we can make into, for conveniently divide into four four groups. One is making the isoprene units by MVA or MEP pathway, or chain elongation, which is most common in most of the living system, and then cyclize, terpene cyclizes these molecules into hydrocarbons, and then the downstream enzymes, including cytochrome P450s, decorate these molecules into final product, including taxol, artemisinin, azadirachtin from neem. So this is how one can classify into this pathway into four stages. So coming to neem, it is a potent antioxidant activity, growth retarding, abnormal bodily development, sterility and toxicity, and it shows at low concentration insects dies. And not many things, if you see the biological activity, only extracts are done. It is shows anti-cancer activity or anti-diabetic activity. If you see the literature, only crude extracts have been seen, no purified compounds. That's where one should purify them and check the biological activity for these things. So once we do that, so if you see that azadirachtin was isolated in 1968 by Morgan, and then partial structure is solved in 68, whereas in 1975, when NMR was used for structural determination, the Nakanishi proposed the structure, but even though it is wrong, more or less it is matched, and the structure was revised in 85 using X-ray and 2D NMR data. And then later in 2007, same Lay and co-workers completed the total synthesis, organic synthesis of azadirachtin A. Then we started working on biosynthesis of these liminoids in neem tree. I don't know how long I will take to complete this pathway, because over 38 genes are involved in the biosynthesis of azadirachtin. So to start with, we take the seeds, kernel, so extract solvent partitions, and then try to get with the different solvents enriched molecules. So this fraction contains nimbin, salinine, that fraction contains other molecules, azadirachtin, nimbin, salinine, azadirachtin B, A, and finally azadirachtin A and B. These fractions were subjected to column chromatography to purify the pure compounds. As you can see that epoxy, azadirachtin, all those pure compounds have been purified and characterized by various spectral studies. So likewise, we started with neem fruit coat, which contains only few major basic liminoids, that is a C ring intact. So we took that fruit coat and then analyzed, followed this extraction method and subjected to column chromatography and purified those azadirone, epoxy azadiradione, and to some extent small molecule, that is gadolinium, which is shown to be potent anti-cancer activities. So then we moved on to the purifying the CC coliminoids, that is a final molecules from neem oil. We took the neem oil, subjected to column chromatography or automated medium pressure liquid chromatography, and then purified compounds were subjected to RB-TRAP or ESI MSMS analysis. So once we extract that, we'll get that chromatogram, and then we try to subject to chromatography. This is just a representative chromatogram. You can see that UPLC based chromatogram, how many compounds are there. So we purified individually each molecules. And then we used MPLC with a large scale purification, and you can see that each fraction was subjected to MPLC and purified all these molecules as shown in below that TLC plate. So then we developed the large scale purification of basic limonades using fruit coat, and we can we can purify up to 10 grams in one run. That's what method we developed, and we can purify how much ever we need in one go. And we publish this data in analytical methods. And all these molecules, you can see that spectrum of like a rainbow. All these molecules, purified molecules, over 30 molecules we purified. And this is a TLC. And we can group them into 3 groups. One is the ring structure is intact. Other one is the ring is gone but not much functionalized. The final one, you can see that so many 8 chiral centers and tetra substituted as a direct DTA. So likewise, these are all green one is final compounds. Once we have all the data, MS and NMR data, all those things, then we moved on to metabolic profiling. So we selected different tissues, flower, leaf, bark, stem, and different stages of fruit till that kernel formation. So we took that and we extracted whole and analyzed by Arbitrum. So we could able to quantify them after drawing the standard graph for individual limonides. So this is a tissue. We will take a tissue and then 0.5 grams and extract using this method. And here we have given the extracted ion and fragmentation. And these are the different stages, total limonides content. We can see that stage 1, 2, 3, 4, flower, stem, bark and all. What is the level of total limonide content based on our 30 limonides? Because those are the major one. Minor one we could not identify, and process is going on for identifying those molecules. So to identify individual limonides, we subject them to Arbitrum with normalized collision energy, vary the normalized collision energy. You see that 15, 20, 25 to up to 40. If you see that 15, you can see that only major M by Z is higher, the over 300. M by Z equals 300. If you increase the normalized collision, so the minor peaks will be, ions will be populated. So we have to use the correct NCE, that is around 25% for the identifying the basic limonides. Whereas in CC-CO limonides, in CC-CO limonides, something went wrong. Anyway, so if you vary the NCE, the different major where only functional groups have been removed, and then finally breaking the CC bonds. So we can get different fragments, depends on the NCE. So we use 20% to get the broad spectrum of M by Z or ions. With this, we establish these are all signature ions. This mechanism we established. If we see these molecules for individual limonides, we can, without any doubt, we can identify this is a molecule present in the crude extract. These are the few representation chromatograms. How we can identify. In the here I have taken a saline. So you can see that bark extract and stem extract, you can see the salicylic is there. How to identify? So we have a standard one, and from this extracted ion chromatogram, we get the molecular ion. With MSMS, we get the fragmentation, and we already have in our library the purified salicylic fragmentation pattern. So this is a purified one, and this is from crude. Exactly matching. If you match this, this peak is salicylic. Without any, beyond any doubt, we can identify each molecule like this. With this, we identified most of the molecules, and we can quantify most of the whatever molecules we can targeted metabolomics. With that, we used, this is a representative chromatogram from flower, stem, fruit coat, leaves, bark, kernel, etc. So if you see that, so the basic limnoids like guanine or azadirachtin are major in fruit coat, whereas the azadiractins are major in kernels, and flowers and all other molecules are present. This really helps when we do the transcriptome of different tissues, where the each transcript expression level, if you identify, at least we can target which enzyme might be involved in the biosynthesis. Also, it helps in the identifying the tissue where what type of molecule we need to isolate. If you see the same graph with individual molecules, with all the tissues, you can see that azadirachtin, stage 4, stage 5, it is highest. It is shifted. Stage 5 and 4, this is highest. Same way, that is azadirachtin B. This should be stage 3. Okay. So stage 3 in saline, stage 3, 4, 5. Likewise, you can see each and individual molecules among all the tissues. We have put a complete data here. So with this C-seco triterpenoids, azadirachtin A, B, and salicylic, nimbein are all those things are major constituents of seed kernel. All the developed stages of fruit coats are mainly containing basic limnoids or C-ring intact molecules. So the limnoid profile of flower and leaves majorly showed the presence of ring intact molecules, including nimosinol and azadirone. So this data is very important in metabolic profiling. So once we have done with neem, we selected all Miliaceae plants and did that profiling. This I have done because the wherever you can see that the azadirone, epoxyazidone, azadiradone, these are basic limnoids which is present in soymida. So if we do transcriptome of that, it is easy to compare with neem and identify the genes involved in those molecules, because neem contains up to azadirachtin. To identify that, this data helps here. So once we have identified all these molecules and purified, so the organic chemist will do the synthetic modification as well as biocatalyst, the microorganisms, whole cell biocatalysis, to see the what type of molecules we get. And then purified molecules we are shared with several groups, and for other biological activity like anti-cancer, anti-diabetic and all, that portion I'll show here. So if you use the mucor species here, you will get two hydroxylations. One is that D-ring and one is that C-ring. So this is the profile you can see, and all stereochemistry, whether alpha or beta. So beta is up, alpha is down. What type of hydroxylation? We use the 2D NMR, HMBC, HSQC and NOC, all those spectral data to identify the stereochemistry of the newly added functional group. So if you just see that, we can modify that. The deacetylation from this, you can get only 17 beta hydroxylated product. Whereas epoxy azadiradione, you will get only 12 beta hydroxylation. So each substrate, small modification, even acetylation, you can tune the what type of product we need or we can get from the uh, microorganism based biocatalysis. Same way we used Cunningham for yeah. So we get two molecules. Those are all characterized by 2D NMR. You can see those two products from salinine. Is sensitive. From basic limonades, we get the hydroxylation single product at 12 position, 11 beta position. The hydroxy group is above the plane of the molecule. So the time course study and pH optimization and temperature optimization has been done. So for these are the molecules. If you see that the R is H, the R is OH. Here R is H, R is OH. The 3 is a substrate we use, 6 is a product we get when we incubate with this microorganism. So when we approached Professor Gopal Kundu, so we tested some of the molecules. They showed potent anti-cancer activity. To trace that where exactly they localize, so we synthesized the tagged, fluorescent tagged molecules. Then we fed with anti-cancer cells. We can see that inhibition of motility wound migration has indicated that the wound migration was seen. And when we do that, the tagging, cellular internalization, you can see different cancer cell lines and no localization observed in the mitochondria. Whereas in endoplasmic reticulum, you can see that the localization occurred. So where they act on it. So with Uday Bandyopadhyay, we gave several molecules and he screened for anti-inflammatory activities. Epoxy azadiradione inhibited the tartamerase activity and found to be so in a reversible, non-competitively and reversible fashion in both in vitro and in vivo. So this can be used, showed a potent anti-inflammatory activity. Similarly, anti-malarial activity with Dhanashekaran, my colleague at NCL, we screened all these molecules. All showed the activity against the malaria. Although chloroquine is a potent compared to these molecules, but these molecules doesn't show the toxicity at this concentration, so that it can be used as a drugs. And he also done the phenotype effect of limonades in cell cycle progression. With this study, the flow cytometry study indicated that except nimbin, all molecules so inhibit the newly invaded merozoites. So they are active against the parasite. If I press, it will go too many. Okay. Okay. So this with Subhash Chandra Bose, he was our colleague. Now he is in Nimans. We gave these molecules. He tested azadiradione, epoxy, and gadolinium against the tau desegregation, where it is involved in the Alzheimer's disease. So the molecules like azadiradione, and gadolinium showed potent disaggregation of the tau protein, aggregated tau protein. So you can see that the fragility, all the molecules, so azadiradione, the aggregates are gone into fibrils. So same
he continued with nimbin and saline, C-columnites, and showed similar activity as observed, and the TEM images are shown behind this slide for saline and nimbin. So with the Ritu Trivedi, we gave these molecules, and she tested against the bone regeneration or osteoblast differentiation and mineralization. So all these molecules, she tested alkaline phosphatase activity. Azadirone had the maximum proliferation at 100 and picomolar and 10 nanomolar. So azadirect A and B shown much better activity in bone regeneration. So we took that forward for a major study. So that we published in different bioorganic medicinal chemistry. This is different. So this shows the similar to that improves the microarchitectural parameters and reduce bone turnover marker in oreoectomy induced osteoporotic mice. So this clearly indicates azadirect A is involved in the bone regeneration. I'll not get into details because I don't know much about me. These are all in collaborative work. I don't know much about these things. So then with Amita Ravikumar, we gave these molecules, and then gadolinium and azadirone shown to be amylase inhibitors. So this indicated that azadiradione and gadolinium from neem inactivated the anti-diabetic target HPA, and may improve to be lead candidates to reduce or control postprandial conditions. So still we need to develop with especially with Subhash and Dhanshekaran. We are modifying the individual molecules and try to get the increase the potency of these modified molecules. That's what we are trying to do that. So next we move to our major work. Sir, how much time I have? I can present full this. If you give full pathway, I'll present. If you give less time. Five minutes. Okay, I'll flash through. You see this. Sorry. These two molecules comes from MVA or MEP pathway. So I'll tell you in later slides which pathway is involved in the biosynthesis of the azadirect or limerides. Once these two molecules formed, it undergoes gernyl diphosphate with condensation of one molecule of IPP with the DMAPP, and then another molecule IPP condensation leads to the formation of farnesyl diphosphate, and then to dimerization to squalene and epoxidation at 2-3 position. This will be S configuration. So leads to the formation of oxidosqualene, which will be cyclized to form the cyclized product. Previously it thought to be eufol or pyrocolol, but we identified this will be the molecule. I'll show you how we identified. And then involved, we also done the identified both the genes to involved in the biosynthesis of millennial, and we up to azadirone we came up here biosynthetic pathway. To deduce this pathway, how much what are the methods we use since biology students, just see that the experiments are simple, but one should do in continuously without the experiment will not wait. Many cases I observe nowadays, if you are really interested, if you start the experiment, you should give complete time to experiment. Now I can see with recent batches where student keep inoculate, sir, I kept in cold room, I'll do protein purification next day. They won't even spin down the cells. So if you don't have time, don't start. But if you start, don't go away from because you keep on repeating. Protein will not degrade because lot of lysozymes are there. protein will degrade. You will not get, sir, I did 10 times. I will not get a protein. What can I do? But you will not tell you kept in the cold room. So the supervisor will be scratching his head. So this I'll always insist, please give the time if research students are here. So as I mentioned, we analyze the transcriptome data. I'll not get into details here. And based on the transcriptome analysis, we identified all the genes. MEP pathway went inside. Anyway, so all MEP pathway genes have been identified, cloned and characterized, and their expression levels are done. Since it is already known in several systems, I'll not give details on those. And then we identified other genes based on the metabolic profiling and tissue specific. You can see that kernel, pericarp, flower, leaf, all transcriptome we used. Tissue specific expression levels, we identified several genes, and we went behind these genes. One by one we went, and experiments are simple. Isolate the RNA, make the cDNA, use the primers, and you will get some band or full length ORF. And then if you don't get, you do the race. And then once if you get the full length gene, you express in yeast or E. coli or whatever it is, and then purify the protein. That purified protein, you will do the assay and analyze the product and characterize. This is the usual procedure what we follow or anyone follow in this area. You see this gernyl diphosphate. We identified clone that expressed, purified the protein and incubated with the gernyl diphosphate. We got the gernial because to analyze by GCGCMS, gernyl diphosphate was hydrolyzed using alkaline phosphatase to get the corresponding alcohol. That mixture we injected in or subjected to GC analysis or GCMS analysis and co-injected. So this is one example. Sometimes what happens in sandalwood, what we faced, even though the co-injection occurs with one metabolite, the fragmentation patterns are different. In that case, we have to purify and characterize and then use a different conditions, chiral column, where they separate stereoisomers or regioisomers will separate. In next enzyme we identified is a farnesyl diphosphate in similar procedure, and same way we identified squalene synthase. Up to here it is a nice thing we went ahead, and cycloatenol also fine. But when we came to epoxidase, how to identify? Because yeast also contains epoxidase. If you put it, you don't know what is this makes. Then we have to add the gene which doesn't produce by the system. So then we choose a yeast, and this we did later, characterization of epoxidase. So before that we try to do triterpene synthase characterization. You can see that there are 6 triterpene synthesis. So the one major one we went behind, AITTS1, and AITTS1 and AITTS2 both are same except some minor changes in the sequences. So this is the expression level in 4 different tissues, and we put it in transformed into cells and then see that what type of compound it makes along with its own endogenous metabolites. If you see that metabolites, you can see that different metabolites along with the assay mixture we injected. The assay mixture gives this product, small peak, which coincides with other metabolites. Now we have to purify that, until unless we purify we cannot say that one. Why we say it is different? Sorry. You see that the fragmentation pattern, these are different. Since they are different, this molecule is different. Now we have to identify. It is not matching with UFOL, it is not matching with tericalol, betarospiral, nothing. So then, how to purify that is such a small compound? We did in large scale, and then the extract was just 120 milligram, subjected to column chromatography, purified, and you can see the TLC, how lanosterol, unknown compound, everything. Just to show here, lot of students, I kept this TLC. You can see that one by one runs if you run the TL column. And this one is a silver nitrate. This we use in the double bond or regioisomer separations. Otherwise, if you use normal silica gel, they won't separate. That's why we use half of this is a silver nitrate impregnated silica gel. Once we got purified compound, using a different NMR data, 2D NMR data, we established that the molecule is Tirucola 724 diene 3 beta all. This is the molecule or intermediate involved in all liminoid biosynthesis. And we patented this and published. And it is not UFOL, it directly goes to Tirucola. So then we came back to squalene epoxide. What we did, we added squalene epoxidase along with AITTS1 so that the expression of AITTS1 product, tirucolol, should go up. That's exactly what you can see that compared to control and with AITTS1, it is small. With the gall promoter and MAF1 means that squalene epoxidase, our overexpression here. You can see that the tirucolol is increased. This indicates that what we inserted, extra squalene epoxidase or squalene epoxidase system, it is active and involved in this squalene epoxide formation. This is exactly in metabolic profile engineering also. The bottleneck enzyme, as I said, mevalonate synthase, which is a bottleneck enzyme. Whenever you want to do that, to get the heterologous expression or production of any terpenoid, you have to make 2-3 copies of that mevalonate diphosphate synthase. HMG-CoA reductase, you have to add 2 or 3 copy numbers. So to just solve again what are the downstream enzymes, we developed that tissue culture facility, and Calyx has been developed in 30 days, and that Calyx we used and we profiled it. So you can see that all the metabolites we identified, because already we developed the method. Based on that method, even Calyx we can identify all these molecules. To put the mechanistic insight, what we did, we fed with 13C labeled glucose. When we fed with 113C, 213C and 1613C glucose into Calyx, we got similar pattern. This is unlabeled mass, this is a labeled carbon. If you expand that, because several lines are there, if you expand, you can clearly see that number of 13C added into M+1. So if you incubate over 20-25 days, you can see complete saturation with 13C. Based on that, we can do the fragmentation pattern. This is a one example salinol. So we already established the MSMS fragmentation pattern and mechanistic and identifying the fragments. Now that knowledge we use to identify these M/Z ions. So now how these carbons, labeled carbons moves, you can see that. With the glucose breaks into pyruvate, glyceraldehyde phosphate or acetyl-CoA. Acetyl-CoA moves with the pathway, pyruvate and glyceraldehyde phosphate moves to this one. You see, both are different labeling. This is very important thing. Both are different carbons. When you add that one, we established this complete labeling studies, and then we went which pathway is involved. When we went through that, mevinolin, which inhibits the MVA pathway, whereas phosphidobicin inhibits the MEP pathway. We use both. When we added mevinolin, no liminoids formed. That means it inhibits the MVA pathway, means no liminoids formation. It indirectly indicates MVA pathway is involved in the biosynthesis. When we added a phosphidobicin, no changes observed with the metabolites. This indicates MVA pathway, MVP pathway or methyl erythritol phosphate pathway is not involved in the liminoid biosynthesis. So then we moved large scale transcriptome analysis. We identified all the genes based on the activity and the structure modification in one-to-one interaction. So we identified all the genes. Now today we have all the genes have been cloned, and we try to characterize them by incubating or transforming in the yeast cell, as well as tobacco, as well as neem. So we identified based on the MS with HRMS. So the three enzymes involved in the formation of millennial. All the three systems we identified. When we co-expressed in neem, what happened? Neem twigs. We dip this agrobacterium infiltration. The twigs are kept in sugar so that they should not die. So when we analyze the products, the higher liminoids, because the moment the millennial formed, which will be pushed into other metabolites which already present in the neem leaves, which we identified in a metabolic profiling. So you can see that increase in the salinine, deacetylsalinine, nimbin, but we could not identify millennial because millennial will not stay. It will further go into other products because biosynthetic pathway already exists in the neem. Whereas tobacco, it won't go. It will stop wherever we keep the enzyme. So still some more is there. I cannot. So we came up to here now. Now we have to move. And this also we identified up to gedulin. We characterized these two genes about 15 days back. Here we came. So this work is going on for analysis of HRMS data. This is how one can do complete natural product work. But one cannot do lot of crosstalk among the groups is required to support. With this, I thank you, and I'll be happy if any questions, especially with the students, if they can ask. I'll be most happy to answer any questions are there. Thank you, sir. So my lab is here in this area, west wing of the main building.
Thank you very much, and I'm sure that I could feel the passion while you were explaining it with your slides itself, that yes, we are doing the experiments. So the passion of that particular experiments are really something, and it reminds me our biochemistry classes, yes, where these particular pathways are being, but the pathways are so important with respect, yes, to each and every, and when you are going from each part of the cells or the time period of this,
Yes.
It's just like a huge expert chemist
Yes.
who is doing so many things, and we are just trying to unravel those things. And because this is an oration lecture, we are not going to have the typical questions over here, but I shall request the students and others to have these questions. So later on during this lunchtime, you can actually have some interaction. But in this oration lecture, normally we don't have these questions. But I'm sure that all of us are going to give a big round of applause to this huge amount of work done by Professor Tulasi Ram, and thank you very much. Thank you.
Hello. Thank you, Dr. H.V. Tulasiram, for wonderfully expounding on chemistry and biology of neem limonoids, the major active principles of the plants. Next, I request Professor Chattopadhyay to inaugurate our website. Please. Thank you, sir. Next, I request Professor Mitali Chatterjee to introduce our honorable speaker, Professor Partho Chakraborty, CSIR IICB, Kolkata, for the Professor K.L. Mukherjee Memorial Lecture. Please felicitate it.
So very good afternoon to all of you, and welcome to this very special session, which is in honor of Professor K.L. Mukherjee, who epitomized the group, the small group of clinician scientists. Professor K.L. Mukherjee was a renowned biochemist, and above that, more than that, what Professor Chattopadhyay also mentioned is if you came to him with a biochemical problem which you faced in the hospital and there was a problem you wanted to identify the biochemical basis, he was always present and ever willing to help people. And what I do recall of him was in his room, which is in the institute where I work at IPGMER, there was a whole cupboard full of Journal of Biological Chemistry, and I had not seen so many JBCs together in the room of a medical biochemist. So that for me was really an eye-opener. But to come to introducing Dr. Partho Chakraborty, it's really my proud privilege and my pleasure to introduce a fellow Matia. We have both graduated and done our MBBS from Medical College. And after that, he continued to do his MD in biochemistry from Banaras Hindu University. He then moved on and beyond doing his MD, also did his PhD, and that was in the US in Boston University Medical School in the US. He returned to India, and I think what is really very nice is that he addressed a problem which is unique to the Indian scenario. And this is something that a clinician scientist is really someone who can pick up. He identified the problem, diabetes. As we know, India is known as the diabetes capital of the world. But more than that is understanding the unique genotype amongst diabetics, which is unique to India, so that, and once you identify that, you also understand that their management is different. So he has been working on metabolic diseases, focusing basically on the metabolic syndrome, and he will tell us about the unique phenotype/genotype that he has been studying. And not only has he been studying the phenotype of these individuals, but also using his biochemistry skills to understand new targets for identifying new chemotherapeutic targets and also using them in the management of diabetes. So with that, I welcome Dr. Partho Chakraborty to take us through the journey and deliver the K.L. Mukherjee Memorial Lecture.
Okay, so I need the point. Is pointer working? Sure. Okay, fine. Thank you very much for inviting me, and it's a real honor. And thank you, Mithali Di, I call her Didi, for this elaborate introduction, and it's a real honor that I've been invited for this K.L. Mukherjee Memorial Lecture. I have heard about him a lot, but never meet him because probably I was too young at the time, but it's really a pleasure to be here. So as Mithali Di has already pointed out that in our lab, we have been working on different metabolic diseases, and of which one is definitely type 2 diabetes, which is, we know the Kolkata data, what we have more than 11% of the adult population of Kolkata are having clinical diabetes. Another 10-12% are pre-diabetic stage. So almost 1/4 of the adult Calcuttans are either diabetic or are on the verge of developing diabetes. But interestingly, what I would like to talk about today, that like any other disease, since we Indians, and particularly I'll show you that we Bengalis are a little bit different genetically in our natural history of our living in the subcontinent, as well as we have some recent historical events which actually shaped our disease profile, our phenotype, and the outcome of disease, and eventually which will implicate in the management of the disease. So, so first thing, oops, this one, yeah, this way only, okay. So you can just go ahead, next, yeah. So for the students, I'll start with this, because this involves genetics, as you know. The natural selection, what happens, though natural forces are there, the environmental forces that shape our the species, and the kind of species will be propagated in a particular environment is selected on the basis of the genetics, right? So we have here in the audience Professor Dwaipayan Bharadwaj, who is one of the pioneers of genetic studies in diabetes in India, and I would say in the world, one of the pioneers here. So this is what we have, but what is more interesting from a clinical standpoint, next please, is this. So apart from natural selection, we do have cultural selection, and India, we have these issues. At the bottom you can see there is a lot of history of migration, there is genetic admixture as well as cultural connectivity that changed and shaped our demographic over the years, over the centuries, and over the millennia. Next, please. So this is a very recent study published recently which showed the genetic diversity in our country because whole genome analysis has been done for approximately 2,000 individuals. And as you can see on the right, so there are different parts of India. They have different genetic makeups, right? And if you look at light blue on the bottom, this scatter plot, you see this light blue is the east, and large majority of these individuals came from West Bengal, and they actually don't fit into the Indian cline, which is marked by these dotted lines, okay? So we have North Indians, South Indians, they are genetically separating. Central India is the middle, but somehow the east and northeast is coming out of this standard Indian cline. So that tells you that we are naturally selected, and so we are a little bit genetically different from the rest of the India, okay? So with this background, we have some recent histories which further shaped our disease profile, and I'll go into that. Next, please. And this is what we call the natural, the cultural selection. And cultural selection of individuals because of human activities shapes our disease profile. And this is one of the classic example happened in US. And you know, one of the darkest human history in the human history is about slave trade, where the West Africans, the black people, have been forced to migrate across Atlantic to the Americas. And what happened during that migration across Atlantic, they were not given any water or food. And most of the black people used to die on the ships, okay? Now question is, who survived? The people preferentially survived who could retain more and more sodium, who could maintain their blood pressure without having a drop of water, okay? So these are people selected, artificially selected by human activity, who has a higher salt retaining potential, right? Now you know America is a land of salt, right? And if you think about French fries, burgers, or whatever, pizza, it's all salty and fatty. So if we just concentrate on salt, so they are consuming so much of salt, and as you know probably, the WHO recommends 2 to 5 grams of salt per day for our consumption. And in one packet of jhalmuri, you know, it's more than 5 grams. So we are also consuming a huge amount of salt in our day-to-day life. And so what happened, these black people in Americas, they are consuming more salt, so the prevalence of salt-sensitive hypertension is huge in US. That means if the blacks they reduce salt intake, their blood pressure is controlled, right? So not so much true for white people. So that's how cultural selection shapes us. And that something happened to India during British era, and particularly in Bengal. Next, please. And this is the history of famines. And this is very important in the context of diabetes, if we want to understand diabetes today. And if you see, particularly in southern, this lower Gangetic plains, there has been huge number of famines, and it is approximately, it's like 1/3 of the Indian population died during last 200 years during British era. And what happens, you can easily imagine the people who survived. So those people survived who could, you know, maintain their life with very low nutrient. Okay, so with little food, people can survive. Those who were more dependent on larger amount of energy, they were perished. Okay, that's what famine did to us. and we are selected in that way. So we are the F2, F3, F4 generations of those individuals who survived with very low nutrient. Okay, so next please. And as you know, for example, this Great Bengal famine in 1946-47, manufactured by Winston Churchill, and there are millions of Bengalis died, and we are their children. And now there are a lot of meta-analysis coming from different parts of the world, including Europe and particularly in Europe and in South America, where we do find there is a significant association of famine exposure with the risk of type 2 diabetes. Now the question is why? The why question is the next slide. Why is this? Because we are the survivors of people who could survive in a very low nutrients condition. But now those days are gone. We are flooded with food with high energy, right? And food is not a source of energy anymore. It's a source of entertainment these days, right? So the major entertainment for human beings across culture is having food together, right? Even if we pass an exam, if we get a good number, we ask our friends, ki khawi bol, right? So that's how we interact with each other, right? So we interact our happy moments with food only. And in this context, it's not about only Western pizza burger diet. Think about classical so-called Indian diet, what we call Indian, and these are super duper bad food. I mean, think biryanis, right? What you have glucose and you have fat. So triglyceride and starch, nothing else essentially. So in this context, the amount of oil we use in last 20 years, the ICMR data shows that Indians have increased their oil consumption by threefold, 300%. So it's a huge amount of oil we are consuming these days. And those all things accumulate in this context of genetics and our recent history. Okay, so next. So what happens? We call metabolic syndrome. Okay, now this is again, the metabolic syndrome is not a disease, it's like a buffet. Okay, so we have multiple issues with our lipids, with our sugar, with our waist circumference, with our heart issues, hypertension, etc. And if you have 3 out of these 5 on the left, this is we call metabolic syndrome. But the disease manifests in some form or the other. So the disease can manifest as hypertension, really high, could be diabetes, type 2 diabetes mostly. For some unfortunate people, it is non-alcoholic fatty liver disease, or now it's called MASLD. Heart disease, cardiovascular events, there are insulin resistance, hyperlipidemia, and for young women, it's oftentimes it's called PCOS, polycystic ovarian disease, which is nothing but an outcome of this metabolic abnormality. So we did a small study with IPGMER, and we found among medical graduates, the girl graduates, 20% of the medical graduates in Kolkata, they are having this PCOD. So this is huge. And I would expect it will not be very different here also. So PCOD is in young women is a major issue as a metabolic disease today. Next, please. So what we know so far? So we know that when we are in 20s, we are having weight average Indian like 50-55 kilograms. When we move to 30s and 40s, it goes to 60 to 70, some cases 75, 80. Nobody is very fat, but in a normal course, we accumulate like 15 to 20 kilograms in our adulthood. And remember, in our adult life, we don't grow any tissue except for fat. Okay? So we essentially grow 15 to 20 kilogram of pure triglyceride in our body, in our adult life, right? And that's what happens. So your body, and as a survivor of famine survivors, our pancreas, which produces insulin and regulates blood sugar, which is quite capable of handling a 55 to 60 kilogram of mass. Now we have 70 to 80 kilograms, and that pancreas has to work hard, and because adipose tissue are insulin sensitive tissue, so it has to work hard to compensate for excess nutrient intake. So what happens with time, beta cell start to grow, there is hypertrophy and there is a beta cell failure, and eventually there is evidence even that there is a death of beta cells. And this ensues a clinical condition called type 2 diabetes, when your sugar goes up, either fasting or postprandial. Next, please. So this is another way of showing the same story. So this is the standard story. This is the European or American story. It's true for almost for, say, 90% of the patients, that with increased weight gain, there is obesity-associated insulin resistance. So where you have higher insulin level, higher glucose level, both, eventually there is a compensatory hyperinsulinemia. That means increase in insulin actually restore blood sugar. And when beta cell or pancreatic beta cell, there is a dysfunction, frank dysfunction. So there is either inability to produce insulin, enough insulin to compensate for the excess sugar load, the patient becomes type 2 diabetes. So that's the standard story. And this story evolves in one individual's life over, say, again, 10, 15, 20 years. Okay? Next, please. So this is another way of putting the same story, that if we put this insulin sensitivity over insulin secretion, we get a hyperbolic curve where an individual lean person goes to obese, and if that person follows the hyperbolic curve, that means if he or she can produce more and more insulin with more obesity, then you don't see any diabetes. So that's why we find a lot of obese people who don't have blood sugar. They have pretty fine controlled metabolic parameters. But unfortunately, those people who falls on from this point B to C and D, when there is a drop in the insulin secretion, that is the state of clinical diabetes, right? So that's the story. Okay. But as you know, like India is a land of paradox, and this is true for diabetes also. And this story again begins from, next slide please, from almost 20 years ago, happened in Pune, from where Dr. Tulasiram came. And it happened in Pune, and this young guy named Dr. Yaznik, Chittaranjan Yaznik, on the right side, and his mentor, Dr. Yudkin, on the left. And interestingly, what they found, both were lean and thin, one white, one Indian. If you see the parameters on top, it's BMI, body mass index, which are same. Means height by weight ratio, it's same for both of them, 22.3, pretty lean and thin. But look at body fat percentage. So average Caucasian whites, they have below 10% fat. We have a lean and thin guy, we have 21%. Even you might assume that I'm a lean and thin guy, I have 19% fat in my body, okay? So what it tells you, that we Indians have more fat, even our newborn, say a 2.5 kg newborn, Indian at 2.5 kg Caucasian, they have different fat distribution, right? So we have more fat to begin with. So we are genetically, epigenetically predisposed to have more fat, okay? And fat is one of the seat of insulin resistance and eventual diabetes. So this is the beginning of a story that Indian bodies are different. So we have different body composition, we have different fat composition, different fat distribution, okay? Next, please. And our history tells us this, right? If you look at the Western sculpture versus our sculptures, ours are pretty nice, beautiful, roundish, right? Sweet kind of sculpture we have, rather than chiseled out, cut, muscular, structure of the West, right? And so we have different bodies, and that is associated with, Dr. Yaznik said, it is associated with low birth weight, There is a fat overflow hypothesis. There is thrifty phenotype. There are genetic predisposition. There are other environmental factors we are talking about associated with maternal malnutrition, which is again associated with famine history of famine, right? So these are all associated. And what we have now, we call it normal weight obesity. So although we have normal weight, we are not fat enough, but we have excess of fat in our body, right? So this is the context where from we started. Next, please. So what we did first, so we looked at the data, what is available with our government of India, and through National Family Health Survey, they surveyed each and every district of India, 640 districts. So, and they measured random blood sugar level and had some anthropometric measurements like height, weight stuff. So what we did, we took each and every district's data and put it on an Indian map. Please, next slide, please. And what we see here, something like that. So this is the prevalence of diabetes in India. It's like 5-10 years ago. And if you look at, the yellow part is more diabetes, green is less. So you can easily see there are two clusters of high diabetes people. Starts from Kolkata on the southern Bengal, across the Odisha coast to Andhra to Tamil Nadu and Kerala. Incidentally, Kerala has the highest prevalence of diabetes in India. And there is another cluster on the north in the Delhi, Haryana, Punjab region, right? So that clearly tells you the more diabetic patients are definitely associated with the kind of dietary habit, which is a rice belt, and that's the aloo paratha makhan belt, right? So two belts we have with very high diabetes, right? Now, interestingly, you know, I just looked at recently Zomato and Swiggy data, and it gives you very interesting perspective. So if you just draw a line from Maharashtra to Sikkim, right, obliquely, the lower part, the majority of the Swiggy Zomato orders are biryani, and the other part, it's pizza, okay? And majority is veg pizza, by the way. And that tells you the dietary pattern and habits are different, so we have very different kinds of diabetes, and right? So this is rice induced, this is, say, makhan induced or whatever. Okay. Now, next please. Now, if we plot the lean diabetes, right, as we are talking about normalized obesity and all this, so the pattern changes. Now yellow is mostly in the central India, right? So we have lean and thin diabetes, like the features like what I have. These patients are mostly located in the central part of India, whereas the obesity-induced diabetes are in the coast and in the Delhi region, right? So the pattern tells you that the histories were different, the genetics and epigenetics were different, and these are probably, they are geographically separated, right? And I can tell you, Dvipendra can actually tell us better, that almost in our data we see almost 50% of the diabetic patients in Bengal, they are in the normal weight category. They are not obese. So we blame obesity for diabetes all the time, but in our case at least 50% are not obese at all. They're very lean and thin. And if you remember my first slide, there's a guy, a lean and thin guy. I gave a picture. He came to our clinic in our cohort. He had a fasting sugar of 350, and I'll tell you that story later. Okay, next. So then when we looked at this data, as I said, they are geographically separated. Now if we cluster it based on the standard anthropometric and biochemical data, we could find at least 4 clear clusters, okay? So 4 different types of diabetes. And as you can see, cluster 2 and 4 marked, underlined in red, the BMI or the body mass index, which gives you a parameter of obesity, they are under 25. That tells you they are not obese. So out of 4 clusters, at least 2 are non-obese diabetes in India, right? This is a national data, and moreover, if you see, we did find a difference of risk of developing diabetes among male and female. And if you see on the right that graph, on the left side you see the males, which are in blue, they are having higher risk when the BMI is low, and after BMI 25, the risk becomes almost equal. So that what it tells you that the non-obese males are at higher risk of developing type 2 diabetes rather than the females. And I'll show you later, and these males are mostly comes from poor background, rural background, and they are not obese people, right? Okay, next please. So what we did after this nationwide data churning, we developed our own cohort in a rural cohort in North Chobbish Pargona, 24 Parganas in Machlandapur area. And we selected few villages and we collected samples. We ran a whole bunch of clinics to identify the diabetic patients and all. And next, please. In collaboration with an NGO. And unfortunately, I had a PhD student who was, again, a Matia alumni. And so I could send him to the villages and have this medical survey. So what we did, so we surveyed and we could select more than 1,000 individuals, mostly women, because in rural setting, women actually respond to medical attention, not the males. And we did whole bunch of biochemistry and imaging study to characterize these diabetic patients. We classified them non-obese and obese diabetes and did some statistical analysis. So what we find, what is important, next please, is this. So we find these are lean individuals. So we are focused on this lean, very Indian type of diabetes. They have very poor response in as an insulin response. So when we give them glucose, okay, in an oral glucose tolerance test, you can see on top graph, both obese and non-diabetes, they have super rise in blood sugar level, which is expected, almost equal. But if you look at their insulin response, the obese people, the blue one, they have an insulin response, which we study in our textbook, that if you give glucose, insulin is produced and to compensate that glucose load. But these lean individuals, they have very, very poor insulin response. So that tells you, we have other studies to support that they do have a defect in beta cell function. Okay, so their pancreatic beta cells are problematic and they don't produce enough insulin in response to a nutrient load. And so the figure, the photograph I showed you, as I told you, so this man had used to drive a cycle van. It's a heavily strenuous work. You have to load, they put like few quintals of load and you have to paddle it manually. And that guy suffered from a injury, road traffic accident. After that, she could not run that cycle van. So what happened? She had a tea stall for last 1.5 years. Okay, so his exercise is gone. Okay, he remains lean and thin. He remains same food habit, and within 1.5 years he developed diabetes. So that exercise that survived him, which is an absolutely almost inhuman exercise he used to do, driving those cycle van, but that kept him alive. But within 1.5 years, he developed this diabetes because probably he had a very low beta cell reserve to begin with. And ever since he started running the tea stall, his exercise stopped and he developed diabetes. So this is a kind of diabetes patients we see in our part of the world. So there is an impairment of compensatory hyperinsulinemia, what I said to you in the earlier slides. Okay. Now in the West also, they have also started to identify these individuals, and now they are saying atypical form of diabetes. And what we have been claiming that 50% of us are these. They have very a poor insulin response, right? And for West, it is atypical. For us, it's a general theme. But again, in the textbook and others, we don't see that presence of this particular disease category, right? Next, please. So we tried to understand what could be the basis for this beta cell dysfunction. So we took some publicly available data of single cell sequencing of type 2 diabetes pancreas and islets, and we looked at the gene expression pattern. And interestingly, the bottom line is what we found, that the lean, these diabetic patients, they do start expressing a lot of genes which are not beta cell specific, but rather they are more of a acinar cell type or some ductal cell type. So pancreas develops in embryo from a single type of cells. They differentiate into other branches. But interestingly, so we do see features of dedifferentiation or transdifferentiation, where we see a lot of genes being expressed in the beta cells of lean individuals, lean diabetic patients. So probably that's why their beta cells are dysfunctional. So we're still working on it, not complete, but that's probably one of the reasons. So we have to look at the development of beta cells in these individuals, and accessing pancreas is almost next to impossible in our setting, so we took data from other places. Next, please. So this is so what we proposed. So from the lean to obese, when a person follows this hyperbolic curve, there are some unfortunate individuals who falls off from this line very early in the process. By gaining 4 or 5 kilogram of weight, they become diabetic, and they become type 2 diabetic, and these are what we call the lean diabetic individuals. Okay, and we have published this data in different journals, and there are some examples here. Next, please. Okay, so there has been a classification. So we call it type 2 diabetes or the adult onset diabetes. Now at least we have 2 types of diabetes, right? One is type 2A, which is obesity induced, and there is a type 2B, which is more relevant to the lean individuals, normal weight individuals, and which contributes to food, lifestyle, stress, and genetics definitely. Next, please. So as I have talked about 2 types of diabetes, 2 types of type 2 diabetes, right? Obese and lean. But so the obvious question would be whether there are many types of them, right? And this idea came from this pioneering work. Next slide, please. Next slide. Yeah. Published a few years ago in Lancet Endocrinology, where they have shown that there are other subgroups of diabetes, and they could identify at least 4 different types of type 2 diabetes in Western population, obviously. Next, please. And so, and ever since there is a huge body of literature coming up showing that there are subtypes and their pathologies may be different, and that's why their therapy could be different, okay? And of which, as you can see here on the right top side, so T2D has at least 4 different. One is called SID, what they call SID, severe insulin deficient diabetes, which is very similar to the kind of diabetes we have described. There is SORD, severe insulin resistant diabetes, which is very well described already, mild obesity related diabetes, and mild age related diabetes, right? So these are at least 4 classification. Next slide, please. And again, coming back to Dr. Yaznik's study very recently, what he had shown, he actually took this model and applied to Indian patients, and he classified it, and you can see in his analysis, 52.79, almost 53% of the Indian diabetes are of SID type. SID means severe insulin deficient. So they are having these type of features, right? And moreover, most of them were males, more than 60%. Females were less. That's exactly what we found earlier, that males are having higher risk of developing this diabetes. Whereas in Europe, you see, it's 26%, compared to where in India it's almost double, right? So we have a very different diabetes pattern, and they need probably different type of management protocol. Next, please. So this is another way of showing this. So even the reviewers have started coming on this subclassification of diabetes. So where we have very high A1C levels with very little insulin resistance, but they have mostly problem with insulin production, the SID one. Next, please. But so why this subclassification and all this is required? Required because the protocol our clinicians follow to treat diabetes is given by either from Europe or from ADA, American Diabetic Association. And if you follow that protocol in our patients, what happens, you see there are two papers, one from India, another from Bangladesh. So in India, it's more than 75% patients remains uncontrolled, okay? That means their H1C, A1C levels remains more than 7 or 6.5, whereas in Bangladesh is 82%. So that means even after so much of discussion, so much of drug development, so much of understanding diabetes, a large majority, 3/4 of the patients remains uncontrolled, right? So there is some problem in understanding our pathology itself. That's what we'd like to emphasize. So what we did, we went the other way around. So we looked at uncontrolled diabetes. Interestingly, in our cohort, again, we followed exactly ADA criteria for therapy. 1 drug, 2 drugs, 3 drugs, like that. And again, more than 70% remains uncontrolled. Okay. So we took these uncontrolled individuals. Next slide, please. And we clustered them. Okay. So because all the diabetics, 70% diabetics are uncontrolled, means almost entire population, and we got 3 different clusters of uncontrolled diabetes. Okay, after 1 year of treatment, 1.5 years of treatment in our cohort in Mochlandapur, and you see that the cluster 3 is most interesting. These cluster of uncontrolled diabetic patients, they have triple therapy. You see the green one is triple therapy, double therapy, monotherapy. Even some started getting insulin within 1 year of diagnosis, and they still remain uncontrolled. And interestingly, most of them, a large majority are males, they are lean and thin, and they remain uncontrolled. So usual metformin, glimepiride, DPP-4 inhibitors doesn't work, okay, for these individuals. Okay, next please. So obviously, this calls for a more in-depth look at the patient's level, as well as a need for personalized medicine. And probably Daipanda will agree that we need to revisit genetics once again under this classification system, and maybe we'll be able to find out new genetic markers and pathological, more of pathological insights into this problem. Next, and this is the thank you slide. So these are my collaborators, Dr. Shatinath Mukhopadhyay and Sujay Ghosh, both from IPGMER, and our surgeon collaborator, Dr. Om Tantia from ILS Hospital, from which data I did not show. We take very obese people undergo bariatric surgery. We take their adipose tissue sample and try to see what's going on there. And Dr. Abhijit Chaudhary, we collaborate for fatty liver disease. And so this is my research team. And thank you very much. So,
wonderful. Thank you so much. On behalf of everyone here, thank you very much for showing us the way forward. And perhaps I'd like to believe that Professor K.L. Mukherjee is somewhere in the audience giving you a pat on the back and saying, this is the way to move forward. This is the way that medical research should go forward, that we identify a problem. It's so beautifully you have delineated a problem which is unique to our country, and it is only people like you who can actually find the solutions and take it forward. And this is to all of you young students, is for you to appreciate, I'm sure you all have appreciated how the problem has been identified and how the solutions are the next step forward. We were short on time, so perhaps the precision medicine can remain for the next time. But thank you very much once again. I think a round of applause for a wonderful presentation. Thank you very much.
Thank you very much, Professor Chakraborty, for your excellent lecture. Now I request Dr. Ashima Lahiri Majumdar, President of the Society, to felicitate our distinguished guest, speaker, rather, Dr. H.V. Tulasiram with memento. Thank you, Madam. I request Dr. Hemlata Datta, Vice President of our society, to felicitate Professor Partha Chakraborty with memento. Again, I am requesting Dr. Ashima Lahiri Majumdar to felicitate Professor Chatterjee, Mitali Chatterjee, with memento because incidentally she was K.L. Mukherjee Memorial Lecture Awardee of Online Seminar 2020. As we proceed, I request Dr. Hemanto Kumar Mojumdar, one of the advisors of our society, to felicitate our distinguished chairpersons with Uttariyo. To Professor Chattopadhyay. Thank you, sir. We are indeed fortunate to have such eminent personalities, and we look forward to an engaging and informative program ahead. We shall now proceed to the poster session following the lunch break, where we will witness the research endeavors of talented individuals contributing to the advancement of human development and understanding of health and diseases.
subsequently at State University of New York in Buffalo. And then in various capacities, he visited different laboratories in US. And for many years, she has been working on matrix metalloproteases in cancer. I think with this brief, I invite Snehoshikta to present her talk. Floor is yours. And organizers are telling that your approved time is 20 minutes.
Thank you for introducing me. Thank you, Shamalda. And at the outset, I pay homage to Sir K.L. Mukherjee and Dr. Uma Ganguly. Coming to SNU, I feel always very homely because here I can find so many of my new known faces. My teacher, colleague, student, so it's really great environment. I feel it's very good academic, and Dhruvoda is going to get the whole credit for it, really. It's very nice. Today, you can see what is my topic. It's a molecule, natural molecule, satawarin 4. This satawarin 4 is a steroidal saponin, and it has very good inhibitory effect, not only in cancer. Other people, they do work and found that it is anti-cancer activity. But we have done work in gastric cancer and under hyperglycemic condition, means under comorbid situation also, satawarin work very well. And the molecular mechanism that I'm going to talk, most of my work is in a particular cell line, which is AGA cell. It's a gastric carcinoma cell line. Next slide, please. So gastric cancer is like other, it's pretty much similar, as it has stage 1, stage 2, 3, 4, and you can see at early stage, if you can detect, it's always good for any cancer to cure. But all those different layers in the stomach, initial layer, mucosal layer, submucosal layer, the red one, muscle layer, serosa. So at stage 4, it affects all those layers, and ultimately those cells are getting invasive in character and move from one place to other, which is a metastasis condition. So we are going to take that particular cell line, which is a cell line, but it's cancer cell line. I'll show you how satawarin really work, inhibiting its proliferation as well. This incidence of this cancer, gastric cancer, is pretty high. It's 8% of all the cancer worldwide. Next one. And you have listened to the seminar in the morning. Diabetes is also a kind of disease which is also rising pretty much. You can see the orange color and red color. From 5% it goes to almost 8% from 1980 to 2014. And it's more prevalent in male. Next. So, next one. So I think of a situation which is a comorbid situation. It basically came into my mind when the COVID came in our, in 2020 or 19. And during that time, you have seen the comorbidity is one of the major problem, and patient couldn't survive. And in that situation, comorbid situation, whether any natural product could be a good help. It's a slide which is very good for a student. In regular normal cell, generally the oxygen is used and ultimately used to, glucose is used to pyruvate and oxidative phosphorylation. But in proliferating or cancerous tissues, what happens, the pyruvate is utilized not only for this phosphorylation, oxidative phosphorylation, where more energy can be gotten, but lactate formation is going on because the cancerous cell utilize glycolysis process more, and that's the Warburg effect.
And next, I will show you that in cancerous situation, how cetaverine is really inhibiting. So these are some of the report that we found, that high glucose basically enhances progression in the carcinoma, the one of the carcinoma, which is cholangiocarcinoma. The another one is human lung epithelial cells. There are also, people have found that high glucose promote invasion, and also in breast carcinoma. And you can see some of the transcription factor and some of the signaling pathway is affected. So keeping that in mind, we also started work in our gastric cancer cell. Next. And as Shamolda also mentioned, I work on metalloprotease for a long time because this is one of the enzyme which cleaves extracellular matrix protein. And this extracellular matrix protein, when it cleaves, the cells getting more invasive in nature. Out of so many protein in MMP family, MMP9 is one of the protein which basically associated to inflammation and also metastasis. Next. This enzyme not only is in active form, it's also in latency form, just like other latent form of enzyme like trypsinogen, chymotrypsinogen. This is also like in its latent form as well as active. The active enzyme, active metalloprotease 9, is really 10 times more active. And a lot of people, they also work on cysteine switch mechanism, because metalloprotease is having that kind of cysteine switch. And next. And you know, this is the hallmark of cancer. There are so many events associated to cancer. And next, some of those which is highlighted, those are where metalloprotease has very much involved in angiogenesis, in invasion and metastasis, in tumor promoting, as well as in immune destruction. And this paper of Hanahan and Weinberg, in 2001 they published paper in Cell. They didn't mention all these 10, but in 2011, after 10 years, after a lot of work in research, they ultimately again show this is what are the different events in cancer. So metalloprotease is involved in some of those cellular event. Next. This is a book chapter from one of my student and myself, what we are trying to show that metalloprotease is involved in different stages of when cancer metastasis is going on, in angiogenesis, in invasion, as well as immune surveillance. Now coming to next one, coming to the particular cell line that we are working on under different concentration of glucose. Next. To tell you the truth, really during that time my laboratory has almost no money, but research we are trying to go on, and very little reagent. So I told my student, why don't you go ahead and culture the cell in different concentration of glucose. So our physiological concentration is 5 millimolar and 25 millimolar, which is in hyperglycemic condition. So this is the different color is different concentration of glucose at different time point. So if you look at 24 hours, 1, 2, 3, that's the third panel, and there you can see that different concentration of glucose, almost cells are okay. And we picked 24 hours for rest of my work. And next. As earlier speaker already talked about, when cells are under stress, they are also produce reactive oxygen species, and that's also true when high glucose concentration is there. The first one is normoglycemic, NG, OC, and the other one is high glucose concentration. Because high glucose concentration, that you can see where Ross generation is at least 1.5-fold high. Next. So when those cells are grown in different time hours, you see the middle panel, normoglycemic condition, what we are doing here, is a confluent cell and we make a scratch. So that's why the gap is there in 0 hours. In 24 hours, the gap is kind of closed. And this is very much true in case of high glucose concentration at 36 hours. That means cells are, the wound area is closed, cells are migrating and invasive in nature. So in high glucose concentration, cells are invasive. Next. And those cells under high glucose concentration, they produce metalloprotease 9. The middle one, the white band that you can see in blue background, and the metalloprotease activity is also increased. 9 is increased more, but MMP2, which is, we call it more like a housekeeping gene, that is remains same, and MMP9 activity is also increased. So this is our initial observation, that high glucose concentration produce more ROS, more MMP9 activity, and also invasive in nature. Next. Next, coming to that particular molecule of my interest is Satavurin 4. Yes, it is from Asparagus racemosus, and this is a saponin. It is used traditionally in Ayurvedic medicine for diarrhea, for dysentery, and stomach ulcer. And also, it's a study shows that it's very much active in healing cancer because it has some anti-cancer activity. But not much of data specifically how it works and mode of action. So we are trying to understand some of its major activity. Next, please. Yes. So we grow cells, AGS cells, and we have added different concentration of Satavurin in normoglycemic condition and hyperglycemic condition. And we have seen that IC50 value at hyperglycemic condition is around 2.4, and in normoglycemic condition is 1.5 micromolar. So it's very low concentration. So this is a good one for selection of drug. Next. Yes. Here, if you see this particular C1, normoglycemic condition versus high glucose concentration, the cell viability is little more, but that is very much inhibited when Satavurin is given, and the concentration that we use little less of IC50 value, which is 2.2 micromolar that we used. And those cells, when we have seen under microscope, that also you can see more number of cells there in high glucose concentration, and that is diminished by addition of Satavurin. Satavurin also has the capability to arrest the cell cycle, and we have used Satavurin along with that flavopiridol, which we use as positive control. And basically, what we have seen, it arrests Z0 to G1 cycle, right? To further understand how it's happening, those cells were also used for protein expression of cyclin D1, which is associated to cell cycle, and some of the kinase, CDK4, CDK2. What you can see, cyclin, the middle one, in case of hyperglycemic condition, the cyclin level is increased, the kinase level is also increased, but addition of Satavurin inhibited the kinase activity, and that kinase activity, CDK, inhibited the RB protein phosphorylation is also diminished, and that's why E2F transcription factor, which basically bind with the hypophosphorylated RB protein that is then detached from E2F, and that's why some of the transcription factor protein started increasing. So basically, Satavurin inhibit those kinase molecule associated with cell cycle. Similarly, the apoptosis that can be done by one of these reagents, which is tunnel assay. And in tunnel assay, the more number of green cell is pretty high, means more apoptosis in case of Satavurin, whereas the 24 hours and 36 hours, most of the experiment we have done 24 hours, but 34 hours, the NG and AG, it's not shown here, but it was pretty much what we have seen for 24 hours, but 36 hours, very high apoptotic cells are found. And next. And the similar scratch assay also we have done in presence of Satavurin, and here you can very nicely see very robust effect in wound healing assay. In high glucose concentration, the cells are more of invasive in character, but Satavurin inhibited very much in wound healing assay, in transwell where we do the cell migration assay, because there are two chambers where cell from upper layer it goes down in the lower, and there you can count the cells. The more number of cells in the lower chamber means cells are migrating, and high glucose concentration more migratory, but Satavurin decreased that, and also colony formation is also decreased. So that means Satavurin works at the level of cell cycle inhibition at G0 to G1 stage, as well as is migratory property and invasive property. Next, what I'm going to show you, my interest, why MMP9 is more, the transcription is more in high glucose concentration. So the transcription factor, which is AP1, that binds to the promoter region of MMP9. So we have next deleted in the full length, the upper one, where you can see the distal part where AP1 bind, the proximal part where AP1 bind, and the MMP9 transcription assay through luciferase we are doing, where you can see the full length under hyperglycemic condition shows very high activity, promoter activity. It's almost 5-fold or so. When we delete it, when there is distal part is not there, and the activity, the promoter activity in high glucose condition, from 1 it become 8. So 8-fold increase, whereas in case of only there is proximal region is only 3-fold. So the distal region where AP1 binds, that is the area where high glucose concentration, the more transcription of MMP9, more activity of MMP9, and that is associated to more invasiveness of the cell. Next. Here you can see the changes in epithelial to mesenchymal transition, because this is one of the major criteria, cells morphology change from epithelial to mesenchymal. Some of the marker of epithelial goes down, whereas mesenchymal marker goes up, means cells are more migratory. From through bloodstream, it goes from one region to other, and that's why the metastasis happen. So E-cadherin, epithelial cadherin, is down in case of high glucose, whereas Satavurin reverse that, and other like bimentin or cadherin, N-cadherin, which is high in case of high glucose, diminished by addition of Satavurin. Okay. Next. It reverse also the MMP9 activity. That you can see very nicely, MMP9 activity, which is pretty high on top panel, the middle one, high glucose, where MMP9 activity is almost going to control level to normoglycemic condition because of addition of the satovarin. While MMP9 is very much increased, MMP inhibitor, which is TIMP, tissue inhibitor of metal protease, is just goes in a reverse direction. So MMP9 activity is going high, inhibitor activity is going down. So the satovarin works at the level of MMP9 as well. And yeah, TIMP1, there is a particular assay which is reverse gymography. There also we have seen the activity of TIMP1. I'm not showing it here, but here is basically the gelatin gymography and the Western blot. Next. So in summary, this particular saponin is very effective in inhibition of AGS cell proliferation at very low concentration, 2.4 micromolar. It arrests cell cycle at G0 to G1 phase, thus preventing hyperglycemia-induced excessive cell proliferation. And it also inhibits the migratory and invasive potential by modulating EMT marker and MMP9 activity. So this could be a good target for treatment of comorbid situation. Next. Yes, this is in summary. In case of high glucose, both the ROS generation, MMP9 activity causing more cell invasion, at the same time the arresting of cell cycle. While satovarin is given, it double short. It takes care the MMP9 activity to diminish as well as to take care the apoptosis as well. Next. Yes, this is a paper on satovarin. If somebody wants, can go through. Next. Thank you very much.
So, you have been known to me for years, but this is a very good talk. But I will say for the students actually, this is an example of what we know about comorbidity and how those comorbidity can be mimicked in settings in a lab. So you can have an idea of it. And also because it's all natural products, so you have a plant, actually in Bengali we call shotomuli. This shotomuli. So many of you know this plant. So these are important saponins actually. We have saponins from ginseng also. We have also in soybean also. We have important saponins for anti-cancer drug therapy. So I will say.
Right.
So,
I started to buying from villages who are selling
that,
and I extracted also. But it very lengthy, time taking, and ultimately I bought it from Sigma.
Okay. So you bought it. Okay. But again,
but isolated compound also worked pretty well.
From Resimo. So that's the important plant actually, which has a lot of saponin. So with that note, and I think the organizer will call Dr. Ghosh because they will hand over some of the certificates to the participants, and then we'll start the last lecture. Okay. Hand over.
But in the meantime, if you have any question, that's why concise my talk, make it instead of 20 minutes, 17 minutes, so 2 minutes you can discuss. Sure. Oh, sub question ekshate. Jumble up so that you don't forget who talked on what subject.
Thank you, Dr. Professor Snehu Shikta Sharnokar. And after the assessment of the honorable judges, we have come to a conclusion for our poster session. There are 3 posters which has been declared as the best poster. So for that, I would like to request Dr. Professor Tulshi Ram to present the certificate on the stage. Sir, please come. So the best poster goes to Shayon Kaur, St. Xavier's College. Are you here? Okay, please come. Yes. And get your certificate. Give a big round of applause to Shayon. Shurja Sharathi Kumar from Maulana Abul Kalam Azad University of Technology. Shurja Sharathi, please come over the stage and take your certificate. Yes, all of you can come. Come fast. Give a big round of applause to them. Okay, and now our very dear Prantika Maiti from Sister Nivedita University. Prantika, you can come with all of your friends, those who have presented the poster. Yes. If you are alone, then you can come alone. Give a big round of applause to her. Okay, thank you, sir. Thank you, Tutsiraman sir, for presenting the poster certificates. Now, I would like to welcome stage the chairperson so that we can move forward.
Coming to the last speaker of the session, Dr. Dipanjan Ghosh from National Institute of Pharmaceutical Research, Kolkata. So Dipanjan, education and research, okay. So Dipanjan was a postdoctoral fellow at the University of Texas, MD Anderson Cancer Center, and then also at the University of Texas Health Science Center, Houston, Texas. So Dipanjan is doing many things. Like he's contributed a great deal to study CRISPR-Cas mediated gene deletion, and also he has some interest on the natural products, and he will be talking about role of beta-glucagonine in combating inflammation by modulating long non-coding RNAs. With this, I invite Dipanjan to present his talk. Dipanjan.
Hello. So good afternoon to everyone. Thank you, sir, for a kind introduction. So I would like to express my gratitude to the organizers, especially Ashima Madam, for the invitation she has given to me to present my work. So presently, as Sarah has mentioned, I am working as an assistant professor in the Department of Natural Products in NIPER, Kolkata. So though my background is the biochemistry and biotechnology, But being a professor at natural product departments, so I have taken up some interest and work on some natural products and their biological activities. So here we'll be talking, with which my lab is working last 6 years, one of the molecules called beta-glucogalin, and I will be talking its role in inflammation and how newly we have found that new targets are coming up, which is not proteins, that's non-coding RNAs, long coding RNAs. So how these non-coding RNAs are related to various diseases and how natural products can modulate that. So first thing is, first we, from the morning, we have the 5 speakers have already talked about various what is natural products. So, and how they work on that. So, natural products, in short, is the small molecules of secondary metabolites produced from any living organism, right? And these natural products, so if you consider, as DSC sir already mentioned in his talk, that in our ancient of what has our literature, or if you consider the traditional medicine, the Indian traditional medicine, way back in 1500 BC, so there are Charuk Samhita, there are Sutsrut Samhita, there are Atharva Veda, they are mentioned in lots of medicinal plants, and they are used in various what type of diseases, right? And Dhanantaris, the then doctors, they had this knowledge, and we have come across generation after generation, and this knowledge has come to us. Now, what we have to do, we have to find out the active components and the mode of actions of these molecules, or all these, how they work. So, so now, if you consider the herbal drug, any herbal drug nowadays you can see, they have lot of made of any natural products, right? And then natural products can be of many types. They can have the flavonoids, they can have the alkaloids, the phenolics, terpenoids, any type. And they are considered to be safe, and they have efficient, they have the biological activities, and they are used for the treatment of various diseases. And now, because of their safety in nature, because of efficacy, that they are coming very much popular nowadays. If you consider when, so I don't know this new generation which is called Generation X, they are familiar or not, but when we were in child, we had some minor problems like cough or something. We were given that tulsi or other things, these honey, all these things are homemade things. Even for Kalmic, all the people, Jaishankar sir already talked about it, androgaphilites is there. We use it for the hepatic protections. It's too much, you cannot, no one will take the second time the Kalmic, is so bitter it is. So neem, as already it has been mentioned, one of the talks. So neem, we take the neem bath, and neem is also for anti-diabetic. So aloe vera is one of the popular, so it has its active component, aloin, helps in the wound healing, right? And curcumin is a compound is present in the haldi. Everybody, every day we use all these compounds, and it has its beneficial effects. And curcumin is one of the compound, it has all types of activity. So it is called pain compound, pan-assay interfering compounds. Okay, so amla, the compound I work on, it comes from this amla, and it has lot of antioxidant molecules. So resveratrol, as we know, quercetin, it comes from red grapes, and in the carrot, whatever the food we are taking, The carrot, it is so vitamin A, full of vitamin A, beta carotene. And the fish oil, it contains the lot of omega-3 fatty acid. Then we have a toothache, then people use the clove oil, which contains the eugenols. And now the probiotics dahi, and those which contains short chain fatty acids and peptides, which improves your gut health. So this is everyday life we use, and they have these natural products already, and we have the beneficial effect we enjoy. Now, many of these natural products didn't come as a drug because many of them are not very specific. They have a pain effect. They can have, not only that, some of them can work synergistically, or many of them can have the good bioavailability, and targeting to a particular tissue, as Professor Mukherjee was talking about, how you can specifically target them, is another challenging for the natural products. But still, we have lots of drugs originated from the natural products. I'm giving a few of them which are very common, and there's reserpine used as against the high blood pressure, morphine, the aspirin is a painkiller, so vincristine, vinblastine, people have already mentioned, and Taxol as anti-cancer drug, and quinine and artemisinin, antimalarial drug, those are already in market. So these have seen the natural product-based drugs are very successful also. So now, these natural products, how do they work? They have many natural products works in many ways. Many of them work through mediating the ROS. Okay? And what is ROS? Okay? ROS is the oxygen-free radicals. It can have various forms. Okay? When I call the ROS, it can be any of it. OH dot, hydrogen peroxide, O2 minus, nitric oxide, R dot, RO dot, anything that is with having the free radicals. Okay? And how do we get this ROS? Okay? We are exposed to various things, right? Either exogenous causes or endogenous factors. So exogenous factors are like UV radiation. You can have the environmental toxins. Okay? Your food, okay, that can give you the very much. Your stress conditions, right? Even exogenous, so there is a dysfunction in mitochondrial electron transport chain. And the enzymes, some of the enzymes have mutations, okay, that can produce a lot of ROS. But we are also fortunate, we are blessed to have a natural antioxidant present in our body. Just like reduced glutathione, the ascorbic acid, the vitamin C, vitamin E, and the various enzymes, SODs, glutathione peroxidase catalyst, those are already present. So we have always a balance between their free radical toxicity versus antioxidant, right? So we have to maintain this equilibrium. So natural product really can maintain this equilibrium. They can take care of the ROS production and bring the equilibrium to the make us safe. All right. So if you have a lot of ROS production, so ultimately this will increase the inflammatory reaction. Okay. So inflammatory reactions is good for the body in such an extent because it can take care of infections, especially the infection from the bacterial infections. Okay. But if you don't take care of prolonged infection, the inflammatory reactions, it can be fatal, give rise to the various inflammatory induced diseases like IBD, like sepsis, even it can give rise to neuroinflammatory diseases or neurodegenerative diseases. Okay? So you have to take care of long-term inflammations. Though inflammation initially is required to take care of the infection. Now, so as I mentioned, so how the natural products tackle the inflammation disease. So first and foremost, as I mentioned, it tackles through taking care of the ROS production. They can take care of the various pathways like JNK pathways, MAP kinase pathways. They interfere there. Ultimately, they stop the activity of the NF-kappa beta and NLRP3 mediated inflammation formations. And this way, the natural products take care of the inflammation diseases or inflammations. So, and the natural products are generally, they activate your immune systems. So, immune modulators. So, this way, it helps us to protect our body against various diseases. Okay, even this way, they can help our system against the inflammations. Now, in our lab, we are working in a natural products that's called beta-glucogallin. This compound is found in amla and pomegranate, and it is a conjugation of the glucose and gallic acid. Okay, so it's called gallotannin. So in normally it is produced in, as I mentioned, in the plant itself, and this is the biosynthetic pathway where you have the EDP glucosyl transferase. They transfer the gallic acid to EDP glucoses. The single step they can make the beta-glucogallin, and the same enzyme can produce another 4 gallic acid compound called pentagallic glucose, and which ultimately breaks down, it can make 2 types, gallotannins or elagitannins. And if you see this structure down on the right, so coriragine is one of the example of the elagitannin, non-hydrolyzable tannin. And this, when you take, it can be in the gut bacteria can make it, and various urolithin forms, urolithin A, B, and others, which has a tremendous benefit to us. So beta-glucogallin has been reported for protection against aging, anti-UV radiations. It can protect us from UV radiations. It can help us from mitochondrial dysfunctions. Okay, it can regulate glucose homeostasis. So many ways it can help. It has a many protective pharmacological activities. Now, we wanted to check this compound, how it protects us from the inflammation. So we made an inflammation. Our inflammation model is very simple. We use LPS and we use ROS 264.7, which is marine macrophage cell line. So if you use the LPS marine macrophage, they give the inflammation. And first thing we checked, so when you treated the LPS, and you see we use the 1 microgram LPS and beta-glucogallin at 10 micromolar, 1 microgram per ml, so LPS. So we checked first how it can protect the raw 264.7 cells from the toxicity of LPS-induced toxicity. And clearly you can see that when you use the beta-glucogallin, it protects the LPS-induced toxicity, protects the cells in for 24 hours and 40 hours from LPS-induced toxicity. So it protects the cells, and then how it protects, then we check the ROS activity. So does it protect the ROS? And in our previous speaker, Professor Mukherjee was discussing about the ROS, and this ROS assay we did by DCFDA and the facts we did. Okay, so whenever there is a lot of ROS, we use the reduced DCFDA. So when it reacts with the reactive oxygen species, and it produces the fluorescent DCFDA, and more the ROS, more the fluorescence. So based on this, you can check this to fluorescence in the flow cytometry itself, and you see in raw cell line in upper panel A, it's a raw cell line. In the lower panel, it's the murine peritoneal macrophage. So in both cases, the LPS-induced ROS is protected by the treatment of beta-glucocorticoid. And the last right panel, it shows, so basically, so we use the mitosox, which actually detects the mitochondrial ROS, specifically the mitochondrial ROS, which is also protected. So we can tell that beta-glucan protects the raw macrophages cells against LPS-induced ROS protection. So now we have checked the effect of beta-glucan on nitric oxide. Okay, and this we can use, you can measure by grease reagent. Very simply, you can check that both raw cell line and murine peritoneal macrophages. So when you treat with the LPS, there is a lot of loss, nitric oxide is produced, which is protected by the treatment of the LPS, beta-glucocorticoid. Now, again, so we check the mitochondrial membrane potential. So if you treat with, give the inflammatory reactions, the mitochondrial membrane potential, it decreases. So when it decreases, so it is easily we can check by using these dye, JC1, which actually, when you have a intact mitochondria, so what happens, mitochondrial membrane potential is normal, the JC1 dye goes inside the mitochondrial matrix, and you can see they make the multimeric form. When they make the aggregates form, they make from the green color, they make the red color, but their monomeric form is the green color. But if you have a decreased mitochondrial membrane potential, this aggregates is not formed. So if you stain the cells with JC1, if you see lot of red colors than the green colors, then cells membranes are fine. And if this ratio changes, the membrane potential is decreased. So from there, it can easily see, even from the FACS assay and from the confocal microscopy, we have seen that the JC1 staining, you see this mitochondrial membrane potential is decreased by LPS treatment, and which is protected by the beta-glucuronide treatment. Then we did microarray gene expression, and we found the genes involved in, I'll finish it. So genes involved in what is called all the inflammatory genes and inflammatory cytokines, these genes are elevated, and beta-glucan can bring them down to the normal. So to prove this, so we check the expression of what is the cytokines in pro-inflammatory cytokines by flow cytometry based bead assay, and you see the expression of IL-6 interferon and TNF-alpha, which are pro-inflammatory cytokines, which is elevated by the treatment of LPS, is brought down by beta-glucuronide treatment. So again, we did real-time PCR to check the pro-inflammatory genes expression level, and we found the INOS, COX2, and these TLF4, MyD88, all the pro-inflammatory, even FAX, FOS, TRAM, all these genes which are involved in pro-inflammatory signals pathway, they are elevated by LPS treatment, and they are down-regulated by what is called BGG treatments. Again, we check the effect of BGG in LPS stimulated NF-kappa beta translocated. So as you know, in NF-kappa beta is situated in the inactive form in the cytoplasm with complex with the ICA inhibitor of kappa beta. But when they are activated, the inhibitor of kappa beta is degraded, and the active form of NF-kappa beta P65 and P50, they translocate in the nucleus. And you see, when you treat with the LPS, and there is more P65 and P50, both you can check the in nuclear localization is more compared to the control, and which has been taken care by the LPS by beta glucuron treatment. So BGG attenuated LPS induced NF-kappa beta translocations in the nucleus. Okay. So next we wanted to effect of BGG LPS in NLRP3 and IL-1 beta expression. So NLRP3, as I mentioned, they produce the inflammasome, and when these NLRP inflammasome is blocked, then IL-1 beta is actually produced, and this IL-1 beta gives the inflammation reactions. So you see the BGG, when you treat with the LPS, there is an increase in NLRP3, and also the active IL-1 beta productions in the cells. So this shows that this is a typical signature of inflammation reactions, and which is reduced by the treatment of the BGG. So then we check the effect of BGG in the sepsis models, and these models in the mice, you can induce the sepsis by giving the intraperitoneal LPS for 5 milligram per kg body weight, and when you treat them with the BGG 40 milligram or 20 milligram per mice. So you see in 40 milligram treatment, you can save the mice and really from their death. And 20 milligram also, they can protect the mice. And from the serum, the inflammatory cytokines also shows there is a protections from the pro-inflammatory cytokines like IL-6, interferon gamma, and TNF-alpha. And we check the various tissues. Just I'm taking one of them, is the liver tissue. You can see the necrosis and histological abnormalities when you treat with the LPS group, and which is protected by treatment with the beta-glucan. So again, it helps, beta-glucan treatment helps the cells to maintain their reduced glutathione level, which is decreased in the LPS treatment. And then we did in silico binding assay with the beta-glucan to TLF4, and gives a very good binding energy of -7 kilocalorie per moles. So this is how the beta-glucocaline works actually. So basically, one way it can block the TLF4 for binding with the LPS, or it protects from the ROS productions, and therefore the activity of NF-kappa beta, and it can also do the inflammasome formations by NLRP3 mediated and IL-1 beta formations. So this way it helps and getting the inflammations, treating the inflammations. So this part of work we have published in International Journal of Molecular Sciences, And also, we use the glucogalin to take the arsenic trioxide induced toxicity, and it is same NF kappa beta NLRP3 kappa. So for the short of time, I stop here because I had many talk about new fascinating things about the LNC RNA. Maybe some other days we can talk about that. Okay, thank you very much for your attention.
So thank you, Dipanjan, for your provocating talk, right? So time is short. So if there is any pressing question from the students in particular, please ask to any one of the speakers already in this session, in the post-class session. Any student has any question? So it seems there are questions from the students. So I think you have? No, from no gray hair person is allowed. But anyway, you are exception.
It is regarding this thing.
Yeah, please, Judge.
Compared with the two fruits which are available in the market of the other fruits. Okay, so that we didn't do it. So they cannot, but what one way we can do it, how much glucogalin is present in someone is still, and also is how much PKPD also viability yield and our under the reason you know Scottish we are started to it. No one thing. The problem is what better than
Okay, so this fairly concludes this session. So I thank all those four speakers for their wonderful presentations. So now I hand over the microphone to the organizers.
Thank you, Dr. Dipanjan Ghosh, for the nice presentation. Now I would like to call upon stage Dr. Professor Ashok Ranjan Thakur, sir. Sir, please come on the stage to felicitate Dr. Professor Parashuraman Jayashankar and Dr. Professor Bishwajit Mukherjee with the memento and Uttoriyo. Let's give a big round of applause. And Dr. Professor Bishwajit Mukherjee. Thank you, sir. Now I would like to call upon stage Dr. Professor Dhrubajyoti Chattopadhyay, sir. Sir, please come to felicitate Dr. Professor Snehoshikta Sharnakar, Madam, with the memento and Uttoriyo. Madam. Give her a big round of applause. Dr. Dipanjan Ghosh. Thank you, sir. Now I would like to call upon stage Dr. Professor Arun Lahiri Mojumdar, sir. Sir, please come to the stage. Chairperson of the committee, Dr. Professor Shamul Roy and Dr. Professor Shantanu Paul to chair the last session. To felicitate with Uttoriyo. And I would like to call Dr. Professor Shantanu Roy Chaudhuri to judge the posters. Only you are present. Other two judges are not present here. That is why I'm not calling. Sushanta Roy Chaudhuri, sir. Thank you, sir. Now I would like to call upon stage Dr. Surjit Singh, Department of Biotechnology, Sister Nivedita University, to give the concluding remarks.
As we conclude, the celebration of the 32nd Foundation Day of the Central Calcutta Society for Advancement of Human Development and Research, jointly organized by the Indian National Science Academy and both the Departments of Microbiology and Biotechnology of Sister Nivedita University, we extend our sincere gratitude to everyone who contributed to the success of this remarkable day. The National Symposium on Combating Human Diseases, Quest for Interacting Natural Products provided a rich academic platform that brought together leading scientists, scholars, and young researchers to address urgent health challenges through natural and bioactive compounds. We have honored to host Dr. H.V. Tulasiram from NCL Pune, who delivered the Foundation Day oration highlighting the chemistry and biology of neem limonoids, offering exciting insights into their therapeutic potential. The Professor K.L. Mukherjee Memorial Lecture by Professor Dr. Partho Chakraborty from CSIR IICB enlightened us on the complexity and the population-specific aspect of type 2 diabetes in the Indian context. During the poster session, budding researchers presented innovative ideas and findings reflecting the energy and curiosity of the next generation of scientists. In the Uma Ganguly Memorial Session, we are privileged to hear Dr. Parshuram Jaishankar, who explored bioactive molecules from Andrographis and Hollerina species as promising candidates for treating cancer, neurodegeneration, and neglected diseases. Professor Bishwajit Mukherjee, who shed light on advanced drug targeting strategies using specialized formulations, emphasizing precision delivery in therapeutics. In the final session, we were deeply engaged by Dr. Snehoshikta Shornokar, who discussed how Shatawarin 4 addresses gastric cancer and hyperglycemia comorbidity through inhibition of MMP9 and EMT markers. Lastly, Dr. Dipanjan Ghosh, who presented on the anti-inflammatory role of the beta-glucogalin, focusing on its regulatory effect on long non-coding RNAs. Each session was marked by stimulating discussions, knowledge exchange, and collaborative spirit. We also thank the opportunity to all the chairpersons and moderators for guiding the sessions with academic rigor and insightful commentary. In addition, we would also like to thank our Honorable Vice Chancellor, Professor Dhrubajyoti Chattopadhyay, for his vision and support in organizing this event at SNU to foster a research environment for both our faculties and to our students. We would also like to thank our very own students who volunteered to serve and made this event a successful one. Lastly, let us carry forward the key insight, foster new collaborations, and remain committed to addressing human health challenges through innovative science. Thank you and best wishes from the Central Calcutta Society for Advancement of Human Development Research and SNU. See you all next year.